Hi everyone, waiting a little bit such that people can join. I think you can hear me. Wait a second, wait a second, guys. Wait a second. Yeah, yeah. Wait a second, Matt. Wait a second, guys. Wait a second. Yeah. So I'm still waiting for you guys to join. If anybody is joining at this particular moment or not. So today we're going to talk about a lot of stuff. So we're going to talk about the whole central dogma. But we first start with the transcription today and then move on to other aspects. Okay. Wait a second. So I'm not seeing many people are joining. Is there anybody who has joined? And just say hi or something such that I know everything is going okay. The connection is good. Come on. Okay. So I can see one person has joined at least. So we'll wait a little bit 5 minutes such that people can join. And at the end, we can talk about it. Talk about what we wanted to talk. Okay. So our first topic is transcription in prokaryotes. Then we are going to talk about transcription in eukaryotes, try to compare those processes because this is the most important part of molecular biology, what they ask in IIT-JAM. We're also going to ask some questions and solve some previous year's questions in today's class. Then what we are going to do is look at the gene expression regulation in both eukaryotes and also in prokaryotes. If it is a prokaryote, then what is going to happen? We're going to look at operons. And as a model, we're going to talk about lac operon and trip operon, two different kind of operons. And in case of eukaryotes, the regulation is more complicated. So we're going to look at several elements, both cis and trans regulatory element. Wait a minute, you don't know what is cis regulatory element and what is trans regulatory element? Don't worry, at the end of this class, this would be totally clear. So we're going to talk about the cis regulatory elements, trans regulatory elements, all of those elements, right? Then what we are going to do is sort of talk about how eukaryotic gene expression regulation takes place. If time permits, we are going to look at histone modifications, nucleosome remodeling complexes, all of these things. And the way I have designed these lectures is by the order of priority. If we talk about IIT-JAM and talk about a priority of occurrence of a topic, I have designed those topics according to the order. For example, lac operon and trip operon are super important, so we would learn about them first. Then we talk about the prokaryotic and eukaryotic transcription. Few aspects are important, not everything is important, and it's impossible to cover everything. So that's why we are going to talk about few aspects of those things. Then we are going to talk about eukaryotic gene expression regulation. They don't ask a too deep question about those things, but it's always good to know. So that's kept at the last part of the class. So with that, I would like to start the class. It's almost 5 minutes that we are online. So what do you guys say? I mean, whoever is online right now. But anyway, whoever is online or would be listening to this podcast, so I would suggest you can, let's say if you're looking for somebody who can teach you eukaryotic transcription, then what I would suggest is you go to the eukaryotic transcription part which is the later half of this video, and you can skip the first video because you might have learned prokaryotic transcription so far so many times. So you can quickly look at it, right? And there would be lac operon, there would be trip operon discussion, and all of these things. So stay tuned. And just to let you know who are the new audience, that I'm also available in Unacademy. I mean, that's why you're seeing my video in Unacademy's page. My courses are live there, so, and a lot more relaxed. There are a lot more students there. We go slow, but we cover everything. The whole concept is being slow but steady. We would win the race. Now, I would like to say it's not a race, but it's a journey. So we'd win the journey, and we would like have our own efforts. So shall I start? So give me some positive indication. Give me some positive indication, guys. I'm waiting for your indication such that I can start. Okay, so let's start. So we know that prokaryote need to divide, right? And that's their job. Their sole goal is to divide and increase their number. So hi, so who is this? Okay, so it's biotechnologist Kundan. Good to meet you. So prokaryotes' goal, especially for a bacteria, is to grow, divide, increase their number, reproduce, and that's all. They have very defined life goals. So in order to grow, they need to do two things. First of all, they need to replicate their own DNA, and that's how the replication is super important for them. And second of all, they need to regulate the gene expression or the enzymes that they need for their whole life. So they need to transcribe their DNA as well, right? So in order for metabolism, in order for division, all of these cases, they need to produce some enzymes. And the way these enzymes are produced is via transcription. So I don't have to tell you the central dogma process, which is going from DNA to RNA to protein. That's what we are learning from our childhood, right? So let's talk about a little bit how things are happening in the prokaryotes. So simply the framework is you have a DNA, from there you are producing the mRNA, and from the mRNA it is translated into the protein. What you have to remember first, that in case of prokaryotes, there is no nucleus. So everything is happening in that cytoplasm-like situation. So your DNA is replicating, your things are getting transcribed, genes are getting transcribed, and at the same time, while the genes are getting transcribed, you're also having the translation process. So everything is happening quite together. For an eukaryotic transcription, things are very different, right? I'll go to that. Okay, so just like any other textbook or any other videos or any other educator, I cannot skip and saying that we have three different steps roughly. So we have initiation, elongation, and termination. So I will take you through this step as we go along this video. So, but I want to also point out few important details that are really important for IIT-JAM or any other examination. So let's talk about initiation first. So the initiation step takes place with the help of the RNA polymerase holoenzyme, the sigma factor, the NTPs, and obviously your template, the DNA, right? That means the bacterial chromosomal DNA. Okay, so first thing first, we'll understand some notations. The particular point where the first base synthesis of mRNA takes place is termed as +1. And beyond that, in the right hand side, you can see it's the downstream, and the upper side is upstream. So whenever you see a number -35 or -10, that means it is upstream or at the left side of this start region in terms of the base pairs or the nucleotide number, okay? And downstream means downside. It would be denoted by plus numbers. So everything or any processes like be it a eukaryotic transcription, be it a prokaryotic transcription, it should start from a starting point, right? And the starting point is nothing but the promoter. In this case, two promoters are highlighted and they have specific consensus sequence, meaning that they are highly conserved. across species. So if you take out 5 or 6 different species of bacteria, you would see the same sequences, more or less they are pretty much same. Though it is shown in upward side for simplicity, but it can be also present in the downward side in downstream region. When we talk about promoters in details, we would talk about it. So first step is the assembly of your polymerase, which is the prokaryotic RNA polymerase, and your sigma factor. Now, what I was saying that at -10 or -35 base upstream, the sequences that you have are pretty much consensus. That means they are pretty much similar in multiple genes or multiple species of same bacteria, right? Okay. So what happens is, if the RNA polymerase can get associated with the DNA by its own. It doesn't need the sigma factor to get associated with the DNA. But there's a problem. Without the sigma factor, after it transcribes a little bit of RNA, what you can see here in the screen, after it transcribed a little bit of mRNA, it falls off, it slips off. In other terms, its processivity is so low. So what does processivity means? So you have heard these terms, fidelity, processivity, all of these things. What does processivity means? Processivity simply means how much you can do at a stretch. For example, you can sit for 1 hour and they do 10 maths. So that's your processivity, 10 maths per hour. So for example, for a polymerase, processivity means how many base pair it can synthesize per minute or per second, per unit time, you might say. So that is their processivity. So the processivity without the sigma factor is pretty low. There are the chances that there could be abortion of that initiation process. So it's called abortive initiation as well. And truncated mRNA is formed, which is non-functional, and obviously they are degraded by the degradation machinery of the cell, right? Okay, but let's say the sigma factor is associated with the RNA polymerase. Now the RNA polymerase can move and they don't fall off, as if its processivity has increased. So there are certain sigma factors specific for specific genes which you don't need to remember. You just need to understand that sigma factor enhances its processivity and reduce the chance or the probability that it would fall from the DNA. Okay, that's all. Now we move on to the process of elongation. And I'm trying to keep it as simple as possible, not getting into too much details, but mostly focusing on the concepts, because in IIT-JAM, most of the cases, they are superficial in terms of asking the question, not too much deep, right? So let's talk about the elongation process. So in the elongation process, what would happen? So the RNA polymerase has a helicase activity, it would unwind the DNA, move forward, and at the same time, using a template, it would synthesize the mRNA, right? So for a moment, for simplicity, we would imagine this RNA polymerase to be this circle. So see, RNA polymerase's helicase activity is unwinding the DNA in a direction that it is moving. Normally it moves in a 5' to 3' direction. And there's a template strand. This template strand is actually serving as a template for these RNA to form, right? And the sequence of this template strand is exactly opposite to the sequence of this RNA is exactly complementary to the template strand. Now you already know the template strand is already complementary to other strand here. So the sequence of the RNA and this coding strand is exactly same. They obviously gives question like this. For example, they give question, okay, if this is the sequence of the mRNA, what would be the sequence of the coding strand? Or if it is the sequence of the template, what is the sequence of the mRNA? So these kind of questions are asked. They won't give you a big base pair, let's say 10 base pair, and they would ask the question, okay? So now the question is few terminologies. The template strand is also known as non-coding strand, okay? No, we'll talk about how to solve numerical type questions and all of these things. We are talking about the basic concepts right now. There would be other classes in these series where we're going to talk about the numerical questions. So first we need to build a base on which we are going to solve these questions. If we don't know the basics, most of the cases the numerical questions are actually class 4 level or 5 level numerical questions, just algebra or just like simple arithmetic. So we don't need to bother about that at this particular moment. We'll get back to it. So right now we understood that, okay, so the RNA polymerase has a coding strand whose sequence is exactly similar to the RNA, except it's DNA, so it won't have the uracil, rather than it would have thymine. Now, the template strand is just complementary to the RNA. Template strand is also known as non-coding strand, right? So exactly the blueprint is present in the RNA, which is present in the coding strand as well. So definitely, while the RNA polymerase is moving, there is a channel which is allowing the NTPs to get in and get incorporated to form the RNA. So let's try to look at how exactly RNA polymerase or a polymerase enzyme works. So whenever you would be a scientist and do some in vitro transcription reaction in the lab, you would see that you need to add magnesium into the solution in order for these RNA polymerases to work in an optimum situation. I hope you guys are still with me. So if the RNA polymerase is getting magnesium, it generally works properly. So how magnesium is important? It turns out inside the RNA polymerase's active site, there are magnesium binding pockets. These magnesium binding pockets actually sort of stabilize the charge on the phosphate backbone and help for, I mean, if it stabilize the charge or attract the charge towards itself, so these center become more prone to a nucleophilic attack. And that is how a new base is incorporated and a new phosphodiester bond is formed. And all these things is happening inside the active site of the enzyme. So you have a clear NTP entry site, you have a mRNA entry site and exit site. The NTP is entered and a new particular site, which is called as mRNA exit site. The mRNA, newly synthesized mRNA, is exiting and protruding out. Okay, so far so good. Now we're going to talk about that what really happens. If we talk about antibiotic known as rifampicin, which is specific to prokaryotic DNA, not specific to eukaryotic DNA. So rifampicin actually binds to the active site of the or beta subunit of prokaryotic RNA polymerase and blocks the transcription process or the elongation process. So transcription cannot take place. So now we come to the termination process. Now termination is actually dependent upon a factor known as Rho factor, which I don't have to tell you because you guys already know. It's a hexameric complex, and what happens is this Rho factor try to strip off the mRNA from the DNA template and free the mRNA, and Rho factor later gets disassociated. This is one mode of transcription termination. The second mode is Rho independent termination, where a hairpin is formed, and the hairpin is actually forming a block for movement in the further. At the end of the hairpin, you have a long A rich sequence, and AU bond is relatively weaker than your GC bond, so that's why the probability that it would be detached from this site. So that is how prokaryotic transcription is terminated. So far so good. You guys give me some positive indication. So we learned how things happen in prokaryotes. Now what we should do is learn how things happen in eukaryotes. So our next topic today is to learn about how eukaryotic transcription takes place. Definitely eukaryotic transcription is bit different, right? So let's see how eukaryotic transcription is different from the prokaryotic transcription, okay? Okay, so eukaryotic cells also need to make many enzymes, and that is how it also need its central dogma processes to go on, right? And the way we see the pictures in the book is somewhere like this. So you have RNA polymerase, sort of like it is polymerizing, taking a sense strand as a template, sorry, sense strand as a coding strand and antisense strand as a template, and it is forming the mRNA. What I want to tell you, since we are talking about the eukaryotic transcription, this particular view is very much wrong and it's incorrect. So what you would be saying, in our books we have this view and we know this view, it is correct. Why I'm saying incorrect? Because we in eukaryotes we cannot imagine transcription happening in this way. Why? Because in eukaryotes the DNA is not alone. It is in format of a chromatin, right? So the initiation is very different. A particular gene which need to be transcribed, let's say a minimum size. So a minimum size of a gene, 1 kb. So we are going to talk about how many nucleosome would be there. At least 5 nucleosome if we consider 200 base pair of DNA is wrapping around a nucleosome. So everything is wrapping around a nucleosome. It's not free DNA and the polymerase is not getting a free chance to polymerize it. So we have to imagine things are ultra complicated in case of eukaryotes. So somebody really need to loosen up the DNA. We don't know who somebody is. We are going to build upon the basics such that we can get to there. But somebody really need to pave the way for the eukaryotic RNA polymerase such that it can get the access to the DNA and start from the promoter and move along the gene body to transcribe, right? So let's see what happens. So if the DNA is tightly wrapped around a particular nucleosome core or histone core, it's almost impossible for RNA polymerase to gain the access. And for simplicity, we're looking at one such nucleosome. Actually, there are many such nucleosomes. They are also organized in a three-dimensional manner, so it's like ultra complicated. But somebody need to actually loosen this up. When it is loosened up, these RNA polymerase now can move along the DNA and transcribe the downstream genes, right? So how does the chromatin configuration is switching? Means like from a inaccessible chromatin landscape to an accessible chromatin landscape. How does that happen? It turns out there are histone modifiers, there are nucleosome remodelers. All of these proteins can modify your nucleosomes or the way nucleosome is interacting with the DNA. And how they're doing it, we have a complete different video or complete different course on that. But they're somehow interacting with the nucleosomes and the DNA and making room for these eukaryotic DNA polymerase to move along the DNA, okay? Now we have eukaryotic DNA polymerase, which is very different from prokaryotic DNA polymerase because we have three of them in eukaryotes, Pol 1, Pol 2, and Pol 3. And you can see their PDB structures, how does they look like. It's a protein data bank structure. Anyway, what we need to understand at this moment is the exam point of view. It's important that all of these three RNA polymerases are there in eukaryotes. So Pol 2 generally transcribes the mRNA, whereas Pol 1 transcribes all the rRNA except the 5S. The tRNA and the 5S rRNA are transcribed from RNA Pol 3. This is a hot favorite question for any MSc entrance exam. Which of the following RNA polymerases transcribe 5S rRNA? Or let's say RNA Pol 1 transcribe 18S, all of these RNA Pol 1 transcribe all of the following rRNAs except the 5S rRNA. So this angle is important, which you should pause and focus. And whenever it comes to enzyme, they're super interested in terms of asking questions. So RNA Pol 2 is way more complicated than the RNA Pol in bacteria. So they have like so many different subunits. Now the prokaryotic RNA polymerase and eukaryotic polymerase, if we compare these two, let's see what are the differences and what are the similarities we get. So just like we have a beta and beta dash subunit in case of the prokaryotic RNA polymerase, which forms the active site, here we have a RBP1 and RBP2 subunit, which forms the active site. So you don't need to understand the bits and pieces and all the details of these subunits, but what you really need to understand that to some extent, the way prokaryotic RNA polymerase and the eukaryotic RNA polymerase work or their functional mechanism, they are conserved, right? And it turns out that if we move down the line of evolution from eukaryotic RNA polymerase is actually derived from prokaryotic RNA polymerases, right? But it doesn't have the sigma factor. Then question is, how does the eukaryotic RNA polymerase gets its processivity? We'll come to that. But what is unique about eukaryotic RNA polymerase is it has a C-terminal domain, or you can think it has a like C-terminal tail. It looks like a tail, it's protrude outside, and the way it is drawn is based on the X-ray crystallographic structure of eukaryotic RNA polymerase. And this tail is so important for several modifications because eukaryotic RNA transcription is happening inside the nucleus, and later on it has to go out. While it go out, it should wear a cap in its 5' site, and it should develop a poly A tail at its 3' end. So all these modifications which are happening is happening very near to the C-terminal domain. So C-terminal domain works like a platform for all of the enzymes to sit on them, okay? So let's see how things work. So first, we have our transcription factors, which are TF2 families. These transcription factor TF2 families are known as basal transcription factors. Okay, what are basal transcription factors? So let me tell you something. Transcription of all of the genes of our body is going on at any point of time. You would say that again. What are you saying? The transcription is going on for all of the gene at any point of time for all of the gene. Yes, I'm telling it correctly. But not too much. It is called a basal level of transcription. Very low level of transcription is going on. Why it is going on? Because this basal transcription factor can always attach. But the level of transcription can increase with the help of some specialized transcription factor, which we would look at. But anyway, the TF2D is the most important one. TF2D has a particular component or a particular domain which is known as Tata box binding protein. Okay, so the Tata box binding protein, I don't have to tell you what is Tata box binding protein, but the Tata box binding protein is a DNA binding protein which binds to the DNA and produce a curvature. And unlike any other DNA binding domain, Tata box has a very unique beta-pleated sheet structure. Other things are mostly interacting with the major groove or the minor groove with the alpha helices. But here the Tata box binding protein is unique that it has a beta-pleated sheet as its interaction. Now it would really bend the DNA at least 80 degrees, and that has particular effect. So it would really cause a curvature in the DNA. So let's see the elongation process. So what happens during elongation is most of these initiation factors or these basal transcription factors fall off. That's why I have grayed them out. So they are actually not going to go along or move along with these particular polymerase. Okay, they are going to fall off the DNA. Anyway, next what we are going to look at how exactly the trigger factor works for the elongation. So who tells it now you should move? So who fires that gun for RNA polymerase to say now you run along the gene, right, and transcribe? So the trigger factor is transcription factor 2H, which is super important to understand in case of eukaryotes. Transcription factor 2H has two activities. One, it has a helicase activity, which is unwinding the DNA in the forward direction, and it has a phosphorylase activity. It can work like a kinase, and it can phosphorylate in the heptad repeats of the C-terminal domain. In the C-terminal domain of eukaryotic RNA polymerase, you have heptad repeats, that means 7 amino acid repeats, which has serine residues in them. Now these serine residues are phosphorylated by TF2H. So hi Shubhendu. So we're going to talk about exactly what happens. Okay, so now the C-terminal domain is phosphorylated. Now phosphorylated C-terminal domain work like a signal that okay, we should move right now. But once it moves, only one thing is attached, the TF2F. The TF2F factor which is an initiation factor, is attached to that. And it moves along with it. And as it moves along, it transcribes, right? It transcribes and form the transcript. Now, just like prokaryotic RNA polymerase, the mode of or the way of enzyme catalysis is pretty same. You have your magnesium plus ions present in your active site, which neutralize or which sort of mask the negative charge on the phosphate group, make it a very good target for a nucleophilic attack. And as a result, what happens? New nucleotide is incorporated, and when this particular nucleophile is attacking the phosphate center, a phosphodiester bond is formed. So this phosphodiester bond is formed in a growing RNA, right? And the RNA protrudes out of the mRNA exit site, and a lot of modification can happen in that mRNA. So this fundamental mechanism of enzyme catalysis is pretty much similar. It is also known as metal ion catalysis. You can clearly see metal ions are so important for catalysis here, right? If you guys do PCR reaction or any in vitro transcription reaction, you would always notice that magnesium ions are super important in terms of your buffer composition. Your buffer is composed of magnesium chloride, and you need to modulate magnesium chloride level for get optimum amplification. So that's why magnesium chloride is important for any polymerase activity. Once they asked a question that which of the following ion is important for the functionality of RNA polymerase or DNA polymerase, it's the magnesium ion. Okay. So now what happens is we have many sequences. Cis sequences means like present in the DNA. Cis means like the same side, like it's present in the DNA. One we have talked about, which is promoter. So promoter is a short sequence around the transcription start site where exactly the transcription starts. And here what happens is the RNA pol2 gets recruited, and there are elements which are known as core promoter. Now, these core promoter binding elements are nothing but basal transcription factors, okay? So this is called the minimal promoter. Its activity might not be very high. So though there are variation in promoter's activity or promoter strength, here the promoter strength is very, very low. So it's a basal promoter, okay? Now what happens? We have another sequence which can be several kb upstream. For simplicity, we have only shown it upstream. It can also present in the downstream direction or even middle of the gene itself. So these are called enhancer region. Now how enhancer region works on the DNA or works on the transcription and modulate the transcription? By the way, before we talk about that how enhancer works, I need to tell you that enhancer not necessarily need to be present in the same chromosome. Forget about the same DNA. It doesn't need to be present in the same chromosome at well. It can be present in a completely different chromosome. And in three-dimensional orientation, a chromosomal segment can interact with the other chromosomal segment. So it's an interchromosomal influence of gene expression. This can also happen. So enhancer is a sequence element which boosts the transcription from the basal core promoter. And not only it can boost the transcription, but sometimes it can also repress. So depend upon what type of molecules it is binding to, such as coactivator or co-repressor, the transcription activity is changed from the core promoter elements. So far so good. You guys are still with me or you guys are falling asleep? So here is a model to show you mostly how a eukaryotic transcription initiates. It's very complicated and there are so many molecules, you don't need to focus on that. But see here, there are so special transcription factors. We'll talk about it later on, but these special transcription factors interacts with the coactivators, and these coactivators actually interact with the RNA polymerase and the basal transcription factor, and they together give a signal for RNA polymerase 2 that you should now move on and transcribe the gene. So this is how it happens. So let me drink a little bit of water and then we'll come back. Okay. Fine. So then what happens? The RNA polymerase starts moving along with this TF2F factor, and what happens is there are several factors such as PTFB, which is also known as the elongation factor and all. They help in this elongation process. Now you can see as a RNA polymerase is moving from 5' to 3' direction, your mRNA is jutting out from the RNA polymerase and hanging from the DNA, right? Now, here I want to tell you that this C-terminal domain is pretty much aligned with the mRNA, and it is pretty much near to the mRNA's exit site in the RNA polymerase. Okay. Now what happens is several modification enzymes are actually going to bind on this C-terminal domain, phosphorylated C-terminal domain. So phosphorylated C-terminal domain is serving like a platform on which several modification factors can bind and do certain modifications on these mRNAs. Okay, so let's see how these factors bind. First of all, you know that eukaryotic mRNAs are, or the small heteronuclear mRNAs, having both exons and the introns, right? So they have very big introns, and in between you have very small exons. Now, the mature mRNA does not have the introns, so the introns some way need to be deleted or edited out. So this process is augmented by alternative splicing machinery. And here I'm just empirically telling you that these are alternative splicing enzymes which are acting upon the mRNA before even the whole mRNA can be transcribed. So let's say three elements, one exon, two exons, and one intron is transcribed at this moment, and these alternative splicing enzyme would start splicing process on these two existing exons and try to bring them together and throw the intron out. So it's happening simultaneously. So imagine how complicated it is. First of all, you have to get rid of the chromosome coil, or the chromatin need to be loosened up such that the RNA polymerase can gain access. That's the first degree of problem. Second degree of the problem is like the RNA polymerase need to move all along the gene. Second is like not only RNA polymerase need to bind in the promoter, there are so many basal factors need to bind into the promoter. Now there are several enhancers which might be even present in other chromosome distant location. They need to interact with the core promoter element and set and give the signal that get set go. As it move along in the elongation step, it's not also easy because somebody consistently need to make the track or make the road on which this RNA polymerase is moving accessible. Otherwise, it would pause, it would stumble, right? Because if the chromatin is inaccessible. So it's an incredibly difficult process. There are also capping enzymes which would form the cap, right? The 5' cap. And it also sits on the C-terminal domain. Now we come to termination. We have looked at all the necessary information and necessary concepts that we need to know about the elongation step. That brings us to the termination step. In the termination step, what happens is there are polyadenylation sites in the mRNA. Now these polyadenylation sites are actually recognized by polyadenylation factors such as CPSF or CSTF. These names might be a little bit important, but let's for a moment try to understand these are polyadenylation factors. They augment this tailing business of eukaryotic mRNA. Now, once this polyadenylation takes place, the end is actually degraded by this RAT1 RNase enzyme. And what happens is like it degrades the protruding a little bit of mRNA. So what happens is mRNA is transcribed and it moves a little bit more than it should transcribe. And then the polyadenylation takes place, and the this tray part is actually deleted by this RAT1 RNase protein. Okay, so that pretty much define our process of eukaryotic transcription. But the eukaryotic transcription is not finished yet. I mean, though RNA polymerase has transcribed the amount it wanted to transcribe, it now in order for translation, it need to move out of the nucleus. And moving out of the nucleus is augmented by several exporters. And we'll look about these exporters, how the export happens on all of these things. Then it need to move where? It need to move to the cytoplasm. Inside the cytoplasm, on the rough endoplasmic reticulum, you have a lot of ribosomes sitting, and these ribosomes are actually working to translate these mRNAs, right? So that is how the portion of the central dogma we have revised. So we have looked at the initiation process, how it is similar and dissimilar with the prokaryotes, what are the factors which are used. We have looked at the elongation processes, then we have looked at the termination processes, and now what we are going to spend our next 20 minutes on is on how post transcriptional processing or co-transcriptional processing happens. That means first alternative splicing, capping, and then the adenylation of the mRNA tail, right? So we are going to look at all of these processes. But before that, tell me, you guys are still with me or not? You guys are sleeping, bored, or what is happening? I cannot feel the josh in you guys. Do interact in this live session. Don't think it as a just boring class. So interact. If you have a question, you try to interact. Okay, so we are pretty much done about these parts. Now we are going to talk about alternative splicing, okay? Or let's say capping. So let's talk about the mRNA capping process. So you guys need to reply. You guys are okay with this pace, or am I going too fast, or we're missing out something? You guys need to tell me something. Come on. Sometimes I go really fast. Tell me, guys, what is the situation right now? You have the Josh left? Come on, guys. It really makes me disappointed. I'm preparing these lectures, waking up in the middle of the night because we have to do a hell lot of work on these visuals, right? So, okay, so people are saying that, okay, slow down. So I'll slow down. I'll repeat one concept multiple times such that it would be easier for you to take it. But I would really suggest that before the day of exam or before the week of the exam, you can also use these videos for your anchoring point, and you can also revise them. You don't need to really read the book throughout because these videos has all of these things. These are kept in mind that you need to qualify IIT-JAM. I give all the necessary information and the concepts that is important. Okay. Okay, so I know the English is some weak for some of you guys, and you guys belong to several places of India. And the problem is my English was also weak, and my Hindi is so weak that if I start talking in Hindi, you guys would run away. So I can't speak in Hindi in that, or I can speak in Hindi, but only in a colloquial manner. So I cannot speak in Hindi for a long time, so I have to give these lectures in English. So please bear with me. Later on, I would suggest you can also change the speed to 1.5x, which would help you. But anyway, let's move back to the 5' capping business. So now the RNA polymerase, you can see it's moving along the DNA, and the RNA is protruding out. Now what is going to happen? This RNA which is protruding out, the C-terminal domain is already bound with so many enzymes, right? The C-terminal domain enzymes are going to act upon the 5' end. So this doesn't happen in case of prokaryotes. Prokaryotes does not get a 5' cap, but eukaryote gets this 5' cap when it is getting transcribed. So that is a big difference between a prokaryotic transcription process versus a eukaryotic transcription process. One gets this capping enzyme while the transcription process is happening. One is not getting the capping business when the transcription is happening. Now the cap is so damn important. We'll come to the importance of the cap. Why you need to put a cap on that? Any idea that why do you need to put a cap on that? Okay, the reason you need to put a cap on that, or this cap is known as M7G cap, is because you need to protect it from nucleus digestion. So the mRNAs can also be degraded by nuclease enzymes, which are present in the nucleus, as present in the cytoplasm. So in next 20 minutes, we are going to look at the mechanisms of the 5' capping, the significance of the 5' capping, because that is important. If we only know about the mechanism and don't know the significance, then it doesn't matter, right? So this video is for practice for anything, like for IIT-JAM, JNU, any exam, what these videos would provide to an insight of the biological machinery or mechanism with augmented visual effects. It would enhance your cognitive skill to remember this topic and understand this topic better, but it is not focused to only IIT-JAM but not JNU, but only IIT-JAM but not any other classes or anything like that. So this is an overall picture, right? Okay. So this is how it works. So now we understand that why we need to have a 5' cap. Because if we don't have a 5' cap, we are in danger. What danger? Because there are nucleases which exonucleases which are going to come from the 5' side and going to eat us, and like eat the mRNA. And if the mRNA is degraded, then there is a big problem. How does the translation would happen? Okay. Wait, let me look at the question first. So somebody is saying that capping dependent upon components. I don't understand the sort of like what question you are trying to ask, but can you rephrase your question a little bit? But I was saying that the importance of the capping lies in the fact that it works like a protection. Okay. Now this cap is nothing but a M7G, 7-methyl guanosine cap. So in this video, we would look at exactly at a molecular level how this cap is synthesized. Definitely you can imagine this is an enzyme-driven process, right? This enzyme-driven process is triggered by the enzymes which is actually sitting on the phosphorylated C-terminal domain of the eukaryotic RNA polymerase. Okay, so this M7G cap has the methyl group at the 7th residue or the 7th position. So that's why it's known as M7 guanosine cap. Okay. M7 guanosine cap not only helps you like protection from the nucleases, but it also helps in actually exporting it out from the nucleus. So let's look at the process in details. Now we are inside the nucleus. So the RNA polymerase is moving in the 5' to 3' direction. As it moves in the 5' to 3' direction, it is allowing the transcription to happen, and the mRNA is protruding out from the enzyme. And now the C-terminal domain is actually a site for modification enzymes, right? So the C-terminal domain can bind to alternative splicing enzyme, capping enzyme, and all of these things which can act upon the 5' cap to modify it. Let's see what happens. So now this mRNA is coming out of this particular eukaryotic RNA polymerase, and at the 5' edge something is going to happen. So sequentially, several enzymes are going to sit on this platform, which is basically C-terminal domain of the RNA polymerase, and going to work on the 5' site of the nascent mRNA, and going to synthesize, build the 5' cap. First enzyme, which is recruited, is known as methyl transferase, triose phosphatase, and guanyl transferase. So all of these enzymes work together hand in hand to form the 5' cap. So let's see how it works. So here is the particular end of the mRNA, right? The 5' end of the mRNA, which is protruding outside. So first thing, triose phosphatase would engage. IIT-JAM, what is IIT-JAM? Okay, so IIT-JAM is an exam for people who are aspiring to do MSc in biotechnology in IITs. Okay, now, yeah, and you have very limited seat, only like 50 seats maybe. And this is people who are aspiring to do their master's degree from IIT. So it's a master's degree level examination for people who are aspiring for IIT-JAM. Okay. Okay. Now we're going to talk about, does that answer your question? Yeah, Saurav? Now first, triose phosphatase, as the name suggests, that it's a phosphatase. If it get rid of phosphate, so it cleaves the phosphate from the 5 prime, right? And then it disengage. After that, there is another enzyme called guanylyl transferase. So the name tells us that it would sort of transfer a guanine group, and exactly what it does. So it uses the GTP and it basically transfers the GTP or the guanine group from the GTP into this growing mRNA 5' end. And later, the last enzyme is going to be recruited, which is methyl transferase. As the name suggests, it would transfer a methyl group at the 7th position. And this is how the M7G cap is formed on the 5' end of the newly synthesized mRNA. So now we pretty much understand how these 3 enzymes work in hand on hand to form the 5' cap, right? Yeah. Now this 5' cap would protect you from nucleases. So in this situation, you can see this nucleus cannot engage to the 5' end because it is protected. Not only that, you would see that M7G cap is super important in terms of translation. When we talk about translation of the central dogma processes, by the way, if you really like this explanation, and if I get enough amount of views and watches, then I would also do a topic on translation and replication as well. But today's focus is on transcription, not too much on the other topics. Okay. So the M7G cap interacts with the poly-A binding protein and form a circle such that many ribosomes can sit on these mRNA and translate from the same mRNA simultaneously. So multiple round of translation can take place simultaneously. So this is another important aspects of the importance of the M7G cap. Other than that, M7G cap helps to export it from the mRNA. Okay, we know mRNA is synthesized inside the nucleus from the DNA with the help of RNA polymerase, but it needs to move out where? Into the cytoplasm, on the endoplasmic reticulum, where it encounters the RNA, so encounters the ribosome. And that is how the translation would occur. But the first and the foremost important step for translation is the mRNA should move out. If the mRNA does not move out the nucleus, translation won't happen. So what happens is that mRNA associates with many of the proteins. None of the name is important apart from the exportin. So exportin helps to export the mRNA out of the nucleus, and then the mRNA, which is having a 5' cap, a tail, can interact with the ribosome and get translated. Now, viruses are super clever. They learn about all the biological processes. So clearly viruses know biology than any human individual. So they are the best biologists in the whole world. So they exactly know how the cell works. First they learn how the cell works and then they hack that. So let's see how influenza virus, which is also in our syllabus, can hack the eukaryotic translation machinery. So normally what happens is, here when the influenza virus gives its particular mRNA, it does not get transcribed because it doesn't have its own cap. If it doesn't have the cap, the host machinery won't recognize it. So it is not getting translated. So what happens? The viral polymerases produce a replica of that, and ultimately it snatches the cap. Okay. Now, while it snatches the cap from the endogenous eukaryotic mRNA and put it on top of it, then the ribosome say, okay, it looks like it is my host mRNA, so I should start translating it. And that is how the viral particles or viral genes are actually translated using the host machinery. So they hack the stuff. So they wore a cap which was not there, and by wearing this cap, our machinery of the host is getting confused that okay, now it is our own body part, we should do it, it's our own stuff, we should translate it as well. That is how they cheat the system to beat our own body. So I hope you enjoyed this part, right? So shall I move on? Guys, I wanted to say, please like the video. Please show your respect by put a thumbs up. Or if you really hate the video, you can also put it down. So give a honest feedback, guys. And do share it with your friends who cannot afford these education but still aspire to apply for several competitive exams or want to learn about biology, because we really make sure that the contents are optimal. So Nilu really joined really late. So we are already halfway through the class, but you can always refine. That's the beauty of these classes, right? Anyway, do subscribe to the Unacademy's live channel, because you would see my videos and many other educators' video live there for just free. That would really augment to give you quality content just for free, and it would definitely augment your learning abilities. I cannot assure that you would get a chance in IIT-JAM or you would get a chance in IIT, but I can assure that you would learn better with these videos or these lectures. So now we move on. As we promised, we would look at the poly A tail process, right? The polyadenylation process. So let's look at how the polyadenylation takes place, right? So we pretty much looked at in the previous video that how does the capping takes place. Now we're focusing on the tail and trying to look at how the tail is actually forming, the poly A tail. Now the question is, what is the importance of the poly A tail, how it is forming, and what if it is not formed, and what is going to happen with the mRNA. So these things we need to keep in our mind while we are reading these things. Okay, so let's talk about that thing. Okay, so moving forward, our transcription, just to remind you, inside the eukaryotes is happening inside the nucleus, right? Okay. NAT questions would be only solved if I get enough amount of positive responses. But for other people who really want to solve or get know how to solve the NAT questions and all of these things, they can always join the Plus platform, and with a very minimal charges. It's only 1,800 rupees per month. That's like charge of watching 2 movies. So these are really easy, right? Now getting back to the concept, we are now inside the eukaryotic nucleus. We see the DNA is getting transcribed, or the gene is getting transcribed by these eukaryotic polymerase. Okay? Okay. Now it is moving in 5' to 3' direction. Okay? Guys, please don't ask questions in the middle of these lectures. So what happens is, like, I lose the track and I lose the flow. So I'll pause after one concept for 5 minutes. This is how we do it from now. We pause after 5 minutes, and then you ask all of your questions. I'll answer your questions. It's not that I don't want to answer your questions, or while I'm teaching, I need to maintain the flow, right? So we are back in the nucleus. Inside the nucleus, we see that in the 5' to 3' direction, the eukaryotic polymerase is moving, right? As it is moving, the mRNA is protruding out of its mRNA exit site, and it is synthesized using the template strand as a replica, right? So using the template strand, the RNA polymerase is synthesizing the mRNA, and there is something called the C-terminal domain of the eukaryotic RNA polymerase, which is getting phosphorylated by several other factors, but the phosphorylated C-terminal domain work like a platform such that many enzymes can attach to it. And we have looked at alternative splicing enzyme, capping enzyme. These things can actually sort of attach to the capping enzyme. Okay. Now there are specific sites present in the mRNA. So let's say this is the length of the mRNA, and there is a specific site. This site is known as polyadenylation choice site, which is AAU-AAA. Okay. Okay, so the AAU-AAA is the polyadenylation selective site. So what would happen is, like, first a factor which is known as CPSF would bind to that. And then another factor, CSTF, or cleavage stimulation factor, would also bind with a little bit distant to that. Okay. Now you can see this scenario in a very simplified view. You have the CPSF either the cleavage polyadenylation stimulating factor or the cleavage stimulating factor at binding at a certain region. Now what would happen? There is a nuclease which would cut it, and there is a polymerase, which is a key important thing. Polyadenylation polymerase, or PAP, polyadenylation polymerase. It would only incorporate polyadenine or adenine into the growing tail. So you have the 3' end, and on top of it, it would keep on adding polyadenylation, keep on adding the poly A tail, right? And that really helps for for helps to move the thing forward. Okay. Now the polyadenylate polymerase would form the tail, and what would happen is this yellow portion is the tail, and the rest of the portion which was cleaved off would be degraded by exonuclease activity. Now this polyadenylation tail is formed. Now polyadenylation tail is actually bound to several proteins which are known as polyadenylation binding protein, or PABP. Later on, when we look at the translation, we would know these sequences are super important in terms of translation, right? Because they interact with the 5' cap and make the RNA or the mRNA as a circle, such that many rounds of translation can happen simultaneously and many ribosomes can work on the same mRNA simultaneously. So the key aspect here, or the key player here, was the CPSF, CSTF, and the polyadenylation binding protein, right? So let's look at what exactly is the benefit of having a poly A tail. Since the poly A tail is already bound with polyadenylation binding protein, so many of the nucleases cannot attack them, and that is why you have a higher lifetime or higher half-life, right? Second of all, ribosome can interact to that and do several round of translation. Translation is augmented. Polyadenylation binding protein also help in nuclear export of the RNA, and definitely, as I have discussed earlier, it helps in the translation process, because polyadenylation binding protein, which is bound to the polyadenylation site, is interacting with the initiation factor of the translation and helping the translation to happen in a circular fashion, and multiple round of translation can take place. Okay, so one particular mRNA can have multiple polyadenylation site, after some exon. For example, this particular mRNA has multiple polyadenylation site marked here in the red. Depending upon which size is chosen, different transcript can be produced. So here 2 different sites were chosen, and as a result 2 different transcript is produced. One of the cases a transcript which is calcitonin, and another cases another vasoactive peptide is produced. So both the cases 2 different transcript is produced, right? One of the cases the calcitonin is produced where the adenylation site which is chosen after the exon 4, whereas if you choose the polyadenylation site after the exon 6, it would form CGRP peptide. Okay, so depending upon which adenylation site you choose, you can give rise to different variety, and this process is used by the immune system to create different type of antibodies or varieties of antibodies, isotypes of antibodies. Okay, so this is pretty much it. And not only that, we know there are membrane bound forms of antibodies and secreted forms of antibodies, and all these membrane bound versus secreted form is determined by your polyadenylation site choice. So that reminds me that polyadenylation site choice is super important for any eukaryotic translation or transcription processes, right? And again, I'm telling you guys, if you guys haven't yet subscribed to Unacademy's JAM channel, you guys definitely subscribe to their channel, such that many of you can get the lectures for free and very high quality education for just free. But if you want to pay, you want to subscribe, you can always subscribe to the Plus platform at very low fees, which is only 1,800 rupees. And if you take a 1-year subscription, it is 800 rupees per month. Cheap as watching 2 movies. Okay guys, so now I'm going to stop 5 minutes. I'm going to take your question answers. So tell me, so what are the questions? So let me just look at it. So first, what is IIT-JAM? So I think I have already explained what is IIT-JAM, and I know you guys right now know what is IIT-JAM, right? Okay, so I don't have to explain that. Then Nilu was asking that, so how we can get ICR Quest? What is ICAR Quest? How can we go get ICAR question papers? But what is ICAR question paper? I don't know what is ICAR question paper. Can you repeat? Okay, so people are saying Nat no. Okay, if you are saying that numerical answer type questions you need to understand, we at the moment do not provide the numerical answer type question in the open platform, but eventually it would be available. I don't know when, but it is now available only in the Unacademy's Plus platform. So you can join there at very low price. You can get my coupon code, which is nothing but AP10. So if you don't know my coupon code, it is AP10, which would give you 10% discount. So that pretty much would tell you about what we need to do. Okay, so yeah, do you guys have any other questions? Tell me guys if you have other question. So now we are pausing for 5 minutes, taking a small break to answer your questions. You need to ask me, like you can ask me anything. By the way, there is a motivation class from 9:30 to 10 p.m. in this same channel, where I would give away all my secrets and tell you 5 top secret hacks that you can use to crack IIT-JAM. And it would be really, really informative and helpful for you guys. So whoever is listening to this podcast can also join to that next podcast, and it would be not long. It would be only 30 minutes. Okay, so if you want to understand about UTRs, we are going to talk about it. The next topic is the 5' UTR and how does it happen. So let's talk about it. So you guys are still with me? Give me some indication in between such that I understand you guys are still there, not falling asleep or not left me. Tell me guys. You guys are still with me or not? So we're going to look at it about all of these things, Nilu. Because yeah, 5' UTRs, 3' UTR, all of these things are very important. And we're going to talk about in next 15 minutes that why they're important. Why they are so important. Okay? So if you guys don't have any more questions, so I should move on a little bit and start this stuff. So right now we're going to talk about untranslated regions. Okay? Untranslated region means what are the untranslated region? So here is a eukaryotic mRNA, which has a 5' cap, 3' polyadenylation tail, and we would think the whole portion of the mRNA has the blueprint for forming the protein. No, that's not true. It looks like the blueprint is a much shorter region, which is marked in blue as well. Now there are some region which is marked in yellow and marked in red are 3' UTRs and 5' UTR respectively. UTR stands for untranslated region. As the name suggests, UTR means like a region which is not translated by the ribosome. So ribosome don't recognize this region. So it is beyond the stop codon. So then the question is, while you are generating the blueprint or decoding the blueprint, why you want to put some extra pages in this blueprint or extra space in this blueprint, right? Why do you want to put some extra place in this blueprint? Is it something important that tells the decoder? Decoder is basically the ribosome, because ribosome actually makes sense what is written in this mRNA code, right, in the codons. So that's why ribosome is the decoder of this blueprint. So does this 3' UTR or 5' UTR has some message to the ribosome how this thing can happen or anything, or do they have other function? So that is the topic of discussion for next 5 minutes. Okay, so in the 5' UTR are generally small, the 3' UTRs are the big ones. Now the ribosome move from like the translate the coding region and get the protein translated, but it does not translate the UTR regions. Okay, Now let's see what UTR can particularly do. UTRs can help in nuclear export of mRNA. Okay, so UTRs can really help export of a mRNA from the nucleus to the cytoplasm, because before the translation can occur, it need to be exported out to the cytoplasm, right? Otherwise, how does the translation would happen? It won't happen. Many of the UTR binding proteins actually interact with exportin, and that is how it gets exported. UTR can regulate translation efficiency as well. Let's see how. So here in the 5' UTR region, near the 5' UTR region, there is a cap, right? Now what happens? The initiation factor, which are in orange, bind to the ribosomal small subunit, and then what? It moves along and scanning for the start site. Whenever it finds the start site, the initiator tRNA binds, and here binds your large subunit of the ribosome, and then they move, right? But let's say this particular small subunit of the ribosome, which is moving and scanning for the start site, finds an obstacle in the 5' UTR. And why this obstacle is there, or this loop is there? Because there was some complementary sequences or repetitive sequences which form a hairpin loop-like structure or a secondary structure in the RNA. And that is why now this particular ribosome is facing a problem or facing a sort of obstacle to move along. Okay. Now the initiator tRNA and the ribosome large subunit cannot get assembled because the start site is not recognized right now, right? Okay. UTRs can also regulate the stability of the mRNA. Translation is really dependent upon how stable the mRNA is. Let's say the mRNA is only available for translation only for, let's say, 5 minutes, or let's say 3 minutes. Then the amount of protein that would be produced would be low. Let's say it is a sort of available, or let's say it is sort of intact for half an hour. Many rounds of translation can take place. Many copies of the protein can be formed from using this blueprint, right? So how fast the mRNA is degrading inside the cytoplasm, because none of the mRNA lasts forever. Yeah, there are some exceptions, but anyway, these mRNAs would also degrade by their own, or by the help of their exonuclease enzymes or decapping enzymes. But what happens is this 5' UTR can really prevent that. So this 5' UTR can bind to several proteins which prevent this exonuclease or decapping enzyme to engage towards the 5' end of the mRNA, right? Okay. So let's see, it would increase your half-life of the mRNA, right? And it would allow more proteins to be translated, because simply the RNA is present in the cytoplasm for more duration. Okay. Now many 3' UTRs, which are bigger in size, they can regulate mRNA stability or the translation rate by binding to microRNAs. Okay, see here. So that answer your question on 5' UTR, Nilu. So how it really regulate the translation, or how it is modulating the transcription or translation processes or mRNA stability. So 5' UTR, how it is regulating, we had a pretty good idea. Now we're going to talk about how 3' UTR regulates this thing, okay? So now we are going to look at the 3' UTR. The 3' UTR can bind to microRNAs. So there are specific processing enzymes which can process the microRNA, and the microRNA can bind to the 3' UTR. Now after that, the microRNA binding and the associated protein which is binding with the microRNA, it could decay the mRNA, allow the mRNA to be degraded, or sometimes it serves like a platform for other things to bind and modulate the translation rate. Okay, so this is really important, that how 3' UTR is regulated. 3' UTR is a very good target for many microRNAs, and it's a hotspot for binding for many microRNAs. Not necessarily all of the microRNAs bind at a time, but some of the microRNAs bind to the 3' UTR, right? It's way bigger than the 5' UTR. 3' UTR can indirectly regulate where it is localized inside a particular cell. So mRNA localization is a very important fact. For example, when a neuron is firing, it has to understand that where it should deploy your mRNA. Think about it. The neuron is firing very high frequency, right, at that particular point of time. Now the neuron doesn't have the particular luxury to transport its mRNA from the nucleus to the cytoplasm, from cytoplasm to the synapses, and sometimes these neurons are actually meter long. So what they do, they make the mRNA, keep it in the synapses, and that's why that gives rise to a local level of translation process. So whenever it is required, the whole machinery to produce protein is there, and it quickly produces some neurotransmitter or some other enzymes which is helping the processing of the neurotransmitter. But let's say the neuron want to also move it to the mitochondria, and it has some role in the mitochondria. So all of these things are actually regulated by the 3' UTR binding proteins. So that is how UTR is super important. So that pretty much summarizes that how UTR, 3' UTR and the 5' UTR are super important for the functionality of the mRNA and how they are transcribed. So in brief, they actually maintain the stability, export, they regulate the translation rate, they regulate the localization inside the cells, and many other factors. So these translated regions, which scientists thought that these are junk regions or not important, turned out to be very useful regulatory elements. So mostly these untranslated regions are pretty much unique to the eukaryotes, not present in the prokaryotes. So that pretty much summarizes all the processes that we need to know for eukaryotic transcription and the post-transcriptional regulation. So this modulation that is happening at the mRNA level is happening after the transcription. So it's a post-transcriptional processing. Okay. So if you get it, now I would pause again, and I'm going to talk about a little bit other stuff. So I was telling that the 3' UTR and the 5' UTR has different size. 3' UTR are bigger than the 5' UTR. It is different in terms of the sequences. If you look at the sequence of the 3' UTR, they have 45% GC content, whereas the 5' UTR has a 60% GC content. In terms of the base composition and their melting temperature and GC content, they are different. Okay. The UTRs play diverse role in the central dogma processes. We have appreciated it, right? So this is the whole motivation that we have learned so far. Now we are going to talk about your questions. So now it is the opportunity for you to ask me some questions, and we wait for 5 minutes again. Tell me. Guys, tell me. I'm waiting for your question, guys. Come on. Any kind of question you guys have, you guys can ask me right now. At this particular point, I would like to say Watson's Molecular Biology of the Gene book is the best book to study about the transcription and all of these things. If you have time, always go ahead and read the textbooks. But if you don't have the time right now, I can assure my lectures are good enough to give you all the necessary information and the concepts that you need to know for any exams. I hope that clears your doubt. So now we are pretty much at the end of our lecture series of today's lecture, Essentials of the Molecular Biology. There would be an Essential Molecular Biology 2 video as well in the coming January session. But what we are going to do is, once microRNA bind to the 3' UTR, one possibility, it might degrade it. One possibility, it might not degrade but recruit other proteins which can interact with many other factors which regulates translation rate and any other thing. So there are multiple different possibilities with the 3' UTR and how microRNA really regulate that thing. MicroRNA can also allow silencing of the genes. So we'll talk about how microRNA and small interfering RNA work, how RNA interference work. So all of these procedures would be actually, we have already talked about it in the Unacademy's Plus platform. If you really want to know about all of these things and don't want to waste your time, you have like at least more than 50 videos about all of these things. And each video is 1.5 hour length. You can watch all of these things. We go really slow. We are going too fast today. I know we are going too fast because we have to give you a lot of things in 1.5 hour of time. But there we talk really slow, analyze all the questions that is important for IIT-JAM, analyze all the questions and so far, so forth, right? And 5' UTR is really important for IIT-JAM's point of view or any other point of view, okay? Yeah, definitely. The change of RNA structure might lead to cancer. There are evidences for that, but this is beyond the sort of like stage of our discussion today, and it's not in the syllabus as well. We can discuss it personally in our other Q&A session. I would be having a Q&A session on YouTube, live Q&A session, where you can really ask me questions and about topics and all of these things. We can create our own agenda and all of these things, right? But let's move back to our own lectures to know about one last process about mRNA processing, which is alternative splicing, right? Alternative splicing, right? So alternative splicing is super important. Okay, so we talk about splicing mechanism. So we know that in eukaryotes, we have both introns and the exons. The exons are really big, so exons are small, the introns are really big, right? Okay, so the pre-mRNA consists of both exon and introns, but the mature mRNA has only the particular exons. Now there could be variety of the exons present in the heteronuclear mRNA. Now specific exons can combine and form different type of combinations. Like here you have like orange, yellow, blue. Here you have orange, green, blue. So using different exons, you have created combinations, and these combinations really increase the number of protein. That also tells us that why we need to really study. When scientists read about this one gene, one protein hypothesis, then what happens? They figured out that okay, if we have X amount of genes, we should have X amount of proteins. But it turns out we don't have X amount of proteins. We have way more proteins than X. So we have like, let's say 5x amount of protein than we have. If we have X amount of gene, we have 5x amount of protein. So why do we have so many varieties of protein, and what is the basis of this variation? It turns out the basis of this variation is actually the alternative splicing. And the splice variants for proteins really change their functionality, their cellular localization, their half-life, and many other things. As we go along the lecture in the last 10 minutes, we would try to identify all of these things. So very important concept for IIT-JAM is like there are 3 splicing groups, okay, based on the type of way you can splice. One group is called group 1 intron, group 2 introns, and group 3 introns. Now group 3 introns are the most important for our own interest because group 3 introns are actually important for mRNA splicing. But group 3 introns and group 1 introns all use a nucleophilic attack mechanism to do all of these things. None of these splicing mechanism requires an enzyme. But the third part requires enzyme not to do the chemical process, but to hold things together. We'll talk about it. So this is how it happens. So you have a branch site adenine, which hits on the uracil on the U1 point, and what happens? It forms a lariat, a loop-like structure. Now this end, which have a free 3' hydroxyl group, now hits the adenine, and the lariat flows away, and we have our 2 exons joined together. So easy, right? 2 consecutive nucleophilic attack, and we have our exon joining together, and the intron, which is shown in gray, is moving out as a lariat. So that is the overall process by which your splicing takes place. Now moving backwards, what you really need to understand why this splicing machinery is there. Here in this splicing machinery, you can see there are several proteins such as U1, U2, U6, U5, all of these things. These machineries actually hold the spliceosomes or the splicing stuff together, because you have to imagine this whole process is happening when the RNA is getting transcribed, right? RNA is like protruding out of the RNA polymerase, and C-terminal domain, all of these enzymes are there, and these enzymes are actually helping in the process of alternative splicing. So everything is protruding out, right? So you really need to understand that how complicated this is. If they don't hold it, this fragment which was protruding out would be flowing away. So that is how in eukaryotes you need somebody to hold it properly, and that works like a substratum or like a platform for these subsequent nucleophilic attack to happen. So first you have this A to U nucleophilic attack, and then this particular 3' open end can work like a nucleophile and hit the other end, and that's how it is joined together, right? And the lariat is moved away, and you have the two exons joined together, and that's how alternative splicing work, right? So that pretty much we can cover in this lectures. We are pretty much not done with alternative splicing. There are more to it. Maybe we are going to cover it in other different lecture. So we are done with the content part. So do let me know in the comment section. Please take 5 minutes of your time and write in the comment section that how do you like these videos? Is the teaching adequate? Give me a positive feedback, and then please share it among your friends who cannot afford education and cannot afford a tuition or all of these things, or not going to a very good college. So this online system would give you college level education for free, right? So with that, I would ask you guys to ask me some question if you have any, or otherwise we can stop the session. We are pretty much at the end of this session. Tell me guys, do you guys have any questions? Tell me guys, it's your time to ask something. You don't need to ask the question in English. I can understand Hindi but can't speak Hindi too well. So you can ask me the question in Hindi. I would answer you in the English in proper way. Or I can also try to answer you in... I'm literally going to take all of the subjects. But sequentially, I don't know when, but yeah, mostly everything. There would be also study with me sessions that would tell you exactly, you open a book and how you can study. So that's how you would get to know what things goes on inside a topper's brain when they study. So all of these things would be there. So tell your friends that there is a particular podcast session from 9:30 to 10 by me. That's a motivational session, and we are going to plan out and give out all the secrets that I have used to crack the exam like anything. So I'm going to give out all the secrets. So it's from 9:30 to 10. Don't miss out that. Tell your friends that they can also join and interact with me, so you have a better interaction in that session. We can take all of these questions to that session as well. With that, I would like to stop the streaming and stop this class. I hope you enjoyed this class. Don't forget to hit that bell icon to get more notifications whenever any one of us is online. And Unacademy is bringing all the educators under one roof, and it would provide all the quality education, right? So hopefully you like this video. Bye guys. I'm stopping the streaming.